Hello, I'm Andrew Freeman, and this is the photocatalytic inactivation efficacy of SARS-CoV-2 in HVAC systems. The recent pandemic has emphasized the need for reliable air purification systems in buildings. There are detrimental effects, such as sick building syndrome, to long-term exposure to harmful particles, such as volatile organic compounds in buildings. For example, if someone is smoking in one room, a poorly ventilated building might recirculate the smoke throughout the entire building. The same concept applies to bioaerosols. This image shows the spread of droplets from a person's sneeze or cough over time. After 1 minute, most of the particles still remain in the air. Gravity begins to pull down on the larger particles by the second minute, but by the fourth minute, the entire area is filled with small aerosol droplets. Therefore, air purification in buildings is of utmost importance to prevent the spread of harmful particles. ASHRAE lists 4 main methods of purification: filters, UV light, ionizers, and photocatalytic oxidation. Filters are perhaps the most recognized air purification method. The HEPA filter is shown on the right. They capture particles from the air by deposition with up to 99.97% efficiency. However, there are a few drawbacks. If you can imagine, the better the filter is, the less it lets through, and that generally also includes air. So there's usually a pressure drop associated with a given rating of filter. Also, filters require replacement over time, and perhaps most importantly, they do not inactivate the harmful particles that they capture. This graphic compares the MERV and HEPA rating systems. A HEPA filter outperforms a MERV 16 filter. Particle sizes between 0.2 and 0.3 micrometers are most likely to pass through these filters, as they are less affected by random motion experienced by small particles and gravity experienced by large particles. The next purification method is UV light. In particular, UVC light, or light with a wavelength of 254 nanometers, is the most effective. This wavelength is capable of biological damages such as cell lysis or the breaking apart of cells. For reference, this is one of the reasons why being in the sun, or staring at the sun, for too long is dangerous to humans. Inside of ducts, UV lamps can be placed in many different configurations, a few of those displayed in the bottom right. However, the light alone cannot slow particles, so they have a very short time frame to do their work, especially in high airspeed ducts. Additionally, this method is not quite effective against VOCs. Next are air ionizers. Ionizers create ions in the air that both weigh down incoming particles and capture them with a strong electric potential. These systems are less popular though, particularly since they create ozone, which is a harmful gas, along with the high power requirements to maintain high voltages. Finally, there is photocatalytic oxidation. Also referred to as PCO or PICO, photocatalysis occurs when a semiconductor or metal, such as the popular photocatalyst TiO2, is irradiated with light, which results in the production of reactive oxygen species, or ROS, that degrade particles in the air around them. This system also has the potential to produce ozone, though at a lesser scale. The material also requires eventual replacement, as the catalysts incur blockage over time. Let's look more into how the PCO mechanism works. When light with sufficient energy, usually UV light, irradiates the material, an electron is ejected into the conduction band, leaving a hole in the valence band. Oxygen and water molecules in the air then react with the electrons and holes, creating superoxide anions and hydroxyl radicals respectively. These reactive oxygen species then move on to degrade pollutants. Since this project focuses on the inactivation of bioaerosols, let's look more into that. The process begins as the species perform violent oxidation on the surface of these cells, resulting in cell lysis. Nutrients then begin to escape the cell, as radical species continue to react. Eventually, genetic material is destroyed and the cell is inactivated. Long enough exposure will lead to cell death and calcification. In reactions with bioaerosols, byproducts usually include harmless compounds, such as water or CO2. The degradation of VOCs has a different mechanism, but lies outside the scope of this project. PCO is a relatively new technique and has typically been used in wastewater treatment and hydrogen production. Since its application to air purification is yet to be fully developed, there are still significant gaps in the literature. For example, aerosolized PCO filter experiments in literature do not accurately reflect realistic duct velocities that would be found in HVAC systems. Realistic duct velocities are usually greater than 2 meters per second, while the majority of existing experiments are below 1 meter per second. Our experiment will help validate PCO filters as a viable air purification method for HVAC systems by working to fill in these gaps, while also particularly focusing on the inactivation of SARS-CoV-2. Our experiment will feature varied face velocities between 0.5 and 2.5 meters per second while maintaining constant temperature and humidity. We will also be using viral surrogates, MS2, V6, and T1 bacteriophages as potential choices. I'll get more into the surrogates after detailing the operating conditions, followed by our test schematic, procedure, and future work. The two primary operating conditions are relative humidity and temperature. Humidity is vital to photocatalysis, as if you recall from earlier, the hydroxyl radical is produced from water. There is no clear consensus in literature about the best humidity level, although it seems to depend on interactions between the photocatalytic material and the pollutant. For our experiment, ambient humidity is sufficient and will be monitored. Meanwhile, temperature has little to no effect on PCO in a realistic setting. It will also be kept at ambient levels and monitored. Since aerosolized COVID in a laboratory is a little bit of a health hazard, viral surrogates are necessary. It is important that these surrogates are slightly conservative to disinfection and inactivation compared to SARS-CoV-2, so as to give a reliable comparison. As a general note, enveloped viruses, like COVID, are the most susceptible to inactivation, while non-enveloped viruses are more difficult to inactivate. SARS-CoV-2 is enveloped with a rather large diameter of 100 nanometers and an ssRNA genome of 30 kilobase pairs. MS2, the most popular surrogate in observed literature, is a non-enveloped bacteriophage of 27 nanometers in diameter and 3.5 kb of ssRNA. T1 is also non-enveloped, though with a diameter of 55 to 60 nanometers and 50 kb of dsDNA. Finally, V6 is enveloped with a 75 nanometer diameter and 13 kb of dsRNA. These three potential surrogates have been used in previous experiments in literature as placeholders for infectious viruses and will allow us to perform tests without worries about infecting the laboratory. Speaking of the laboratory, our test setup consists of a 2x2 by 50-foot duct in Herrick Lab. The system has temperature and humidity control, as well as differential pressure sensors and duct velocity control. Additions for our experiment include a nebulizer for aerosolization of the surrogates, the air purification method, followed by a biosampler array and ozone sensor. As the fan pushes air through the system, pitot tubes measure the air speed. A nebulizer then sprays a certain volume of virus particles to be mixed into the airstream. The particles reach the air purification method with differential pressure sensors on either side. Then, the biosampler collects a certain volume of air to be analyzed for residual virus particles. The ozone detects any ozone potentially created by the PCO methods. Transitioning to the top view, here you can see how the biosampler will have 3 collection ports. This will allow us to average measurements across the 3 devices to get a more accurate reading. The project timeline consists of literature validation, preparations, followed by experimentation and data analysis. For a given surrogate, each air purification device, including natural decay, a HEPA filter, UVC light, and PCO filters with and without UV light, will have triplicate trials for each air speed. While the Herrick laboratory is currently undergoing upgrades, our preparations for testing include the implementation of sensors, a detailed CAD schematic, and further literature validation, such as PCO filter coating methods and UV placement and irradiation. In conclusion, photocatalytic oxidation is a new technology for air purification, and current literature is not an accurate reflection of its implementation in HVAC systems. Our experiment intends to help bridge the gap in literature with a standardized HVAC PCO filter testing method and help establish the reliability of PCO systems for viral inactivation and disinfection. Our overarching goal is to reduce the spread of pathogens such as COVID and make buildings a safer and healthier place for people to exist. Further advancements for this project would include monitoring the effects of varied humidity, applying different challenge agents, including volatile organic compounds, implementing non-filter PCO reactors, and analysis of secondary reactions and byproducts in PCO reactions. Thank you all for listening.